Good morning. My name is Vishal Joshi, and today we will be talking about predicting function in UniProt using rule-based and natural language models. Before we proceed, I would like to thank the UniProt consortium comprising of MBI UK, Swiss Institute of Bioinformatics, Switzerland, Protein Information Resource in 2 sites in USA, Washington and New York. UniProt knowledge base provides the scientific community with a comprehensive, high-quality, freely accessible resource of protein sequence and function information. One of its databases is the UniProt knowledge base, which contains expertly reviewed SwissProt entries and unreviewed Tremble records. Reviewed records consist only of 0.2% of UniProt knowledge base because expert curation is time intensive and mainly focuses on model organisms. And that's why we would see that SwissProt is around 569,000 protein entries, while UniProt is 249 million, which is almost 5 times or maybe more of SwissProt. The number of unreviewed records are constantly growing, and a large portion of these do not have experimental data. So here we present 3 automatic annotation systems used by UniProt, which are UniTool and Arba, while the recent and the latest method, which was introduced in UniProt 2204 last year, is ProtLM, which was developed in collaboration with Google DeepMind, which we'll talk about later. UniRule. UniRule system consists of manually annotated rules that specify functional annotation and the conditions which must be satisfied for them to be applied, such as taxonomic scope, family membership from Interpro, and the presence of specific sequence features. Arba, on the contrary, is a fully automatic system. Its full form is Association Rule-Based Annotator, and it's a multi-class learning system based on knowledge learned from SwissProt using frequency item set mining techniques to generate concise annotation models with highest coverage for annotation based on the properties of Interpro group membership and taxonomy. Both these rule-based methods are fed to a rule engine, which take protein sequences, that is SwissProt, to learn from, and Tremble to apply the predictions on, taxonomy data from NCBI, Interpro scan results, which are provided to us by Interpro, which gives us the mapping between Interpro signatures and the UniProt KB records. The rule engine will apply these rules and generate predictions, which will be applied to Tremble mainly. ProtLM is the latest method UniProt KB has introduced, and it has been developed with the groups of Max Pilecki and Lucy Caldwell at Google DeepMind. ProtLM stands for Protein Natural Language Model, and it predicts names for UniProt KB Tremble records, which had earlier the name uncharacterized protein. That means our methods could not provide a meaningful, concise protein description for those Tremble records. ProtLM is trained on UniProt 2021-02 data, and it uses T5 framework. It's a transformer model which takes input as a string and gives output also as text. For our use case, it takes sequence as an input and generates the protein name as an output. But it has the capability to use sequence and maybe provide predictions for protein family, EC numbers, etc. After the UniProt was introduced, we also sampled the predictions which Prot T5 gave, Prot NLM gave, and after sampling, we could find some unsuitable names which were mostly concentrated in between 0.0 and 0.2 confidence score provided by the model. We therefore applied a confidence threshold of 0.2 and above before applying these predictions in UniProt knowledge base. We also started last year a UniProt metal binding challenge to bridge the gap for missing Tremble entries which do not have metal binding. As you can see from this pie chart, there's only 3% of UniProt KB Tremble which has metal binding line types. The challenge was started in April 2022 and was closed in May 2023 this year. We are currently evaluating the results using the information theory, which is based on Bayesian probability. The paper which was